What I've been asked to tell you about is a bit about how the brain works, and particularly about the way in which it controls movement. So I'm just going to give you a very brief introduction into some of the core aspects of how the brain's motor system works. And I guess it's up to you really to decide whether or not this is something that's useful and might inform understanding of BFRBs. So this is the organ that we're interested in, the brain, and I've got a few different brains up here. So starting off with the sort of basic mammalian brain, something that we think probably looked a little bit like a rat brain, moving to a primate, so a monkey. You can see this is actually a macaque monkey. I think macaques were in some of the images that Claire showed earlier on, and then a human brain on the right-hand side. And we know that the common ancestor that we have with rats lived sometime around about just after the end of the era of dinosaurs, so about 65 million years ago. So a lot has changed since then, and there are some special features of primate brains of monkeys and apes and humans that are really quite different to that basic rodent pattern. We're really quite different even to these macaque monkeys. We diverged from them about 23 million years ago, so a long time ago since we last had a common ancestor, but there's perhaps a bit more that we can learn from them about how our motor systems work. So what I've zoomed in on here in this picture is a particular part of the brain that's shown in red. It's called the motor cortex. And we've known since sometime in the middle of the 19th century that this is a very important region for controlling movement. So if we zoom in on this region in the human brain, for example, if I flip this picture of the brain around, so now that red area is more towards the right-hand side, I've done that so it can match up with this image of the motor cortex that's taken from a study in the 1950s conducted in Montreal by Penfield and Rasmussen. And this is showing us that red area there is the same as the area that you can see here. So this black line here is the same as the black line here just next to the red area. And that is a very important groove in the brain, or sulcus. It's called the central sulcus. And this motor region here, shown in red, is the area that's just in front of it. And what this picture is summarizing, it's a very old image now, but it's showing us that if you electrically stimulate that area of the brain in patients during the course of a surgery, then you can induce movements of different parts of the body. So, for example, this area that's outlined with a continuous black line, that leads to tongue movements. You can see that there's an area that's highlighted in this dotted line up here. It's a bit higher up, and if you stimulate that, that leads to hand movements. So you can do experiments like these with people while they're awake and conscious. To stimulate the brain in this way isn't painful. The painful thing is making the opening in the skull in order to get to the brain, but actually stimulating the brain itself is something that can be done while people are aware and conscious. And so that's some of the evidence then that we know about that has led us to think about this as a brain region for controlling what we do, for making movements. How does it do that? Well, if we zoom in and look at it, this is now what we're doing is we're looking at this same brain area, but we've zoomed in to look at it through a microscope. And what we can see are lots of different brain cells. So all of these dark black patches are brain cells, but there's some really big ones here. And these were first noticed by a Ukrainian neuroanatomist working in Kyiv in the 1860s. His name was Vladimir Betz, and the cells are called Betz cells after him. And they're really big cells because they have to do something very special. This is what a brain cell looks like if we zoom in on it. So Here's the center of the brain cell. Here are its dendrites. These are the bits of the brain cell that get connections and instructions coming in from other brain cells. And then here is the axon. This is like the output channel for the cell. And this allows that cell to connect up to another brain cell and send signals and instructions to it. But these cells are so big because they have to support axons, these output devices that are absolutely huge, that go all the way, not just to another brain cell just a few millimeters or centimeters away, but they go all the way outside of the brain, all the way down into the spinal cord. So they might be 1 meter long. So something absolutely enormous when it comes to the dimensions that we would normally be thinking about when we're talking about individual cells. And they do that because they need to send instructions to the spinal cord, and then from there the instructions go to our muscles to actually make a movement happen. So we know that these cells here are what ultimately produce movements. So that's a little bit about how we know that movements are actually being executed by the brain. But how does the brain know which are the right movements to make at any time? And the brain gets its movements made by the motor cortex shown in red here. But the motor cortex gets its instructions from the region that's shown in blue here. This is the premotor area, or PMA, as it's been indicated here in these pictures. So you can see that humans have a big premotor cortex, so do macaques, and there's even a small one in the mouse here. But there's something that happened in evolution whereby this area really began to change a lot in monkeys and apes and humans compared to other mammals. So we're actually only looking at one part of this premotor cortex, and we'll see a bit more of it in a minute. And it turns out that one of the important primate innovations that monkeys and humans have is multiple premotor areas that are all sending different types of instructions to the motor cortex, telling it what to do. So how does it do that? Well, I'm going to give you a very brief example. If you were driving here this morning, or if you were walking here, the chances are that probably at some point you came to a traffic light. So you might have been looking at the traffic light at a green light, telling you to press the accelerator and move forward, or at the red light, telling you to press a brake and to stop moving. And we know that learning to map different instructions or stimuli in the world onto different actions is something that blue area, the premotor cortex, does. So we know that from experiments conducted here in Oxford, where people have looked at how people and animals can learn to make associations between just about anything, any visual stimulus, and any action. You can learn to do one thing when you see a green light, another thing when you see a red light. And that ability depends on this premotor area. So turning information out there in the world into an action is something that this brain region does. And one of the great things about primates is that they can learn all sorts of arbitrary mappings. There's nothing predetermined in the world about yellow or red, or green or blue, or any other colour that should link it to stopping or starting. But we have a brain that's ready-made to learn these arbitrary associations. So that's one of the things that the premotor system does. But now we're going to focus in on it in a little bit more detail in these premotor areas. So now we're still looking at that same brain area. There's that motor cortex. It's changed colour. Now it's yellow here on this picture. And then we're going to zoom in and look at the premotor areas that are sending these instructions in a bit more detail. So they're actually divided up into different parts. A dorsal part, the blue area here. This is the one that's really interested in learning these arbitrary associations that allow us to do all sorts of clever things. But it's not the only part. So primates like us have multiple other areas. that are sending all sorts of other instructions. There's this green ventral premotor area here. And now on the next slide, what we're going to do is we're going to turn the brain round, because here what we're doing is we're looking at the brain from the side, but there's a whole other surface of the brain, like the sort of dark side of the brain that you could see if you were to look from the middle outwards. And here you can see that there are other areas. These are called the cingulate motor areas of the brain. They're called that because this word cingulate comes from a word that's linked to a word for belt, or it's cognate with the word cinch as well. And the idea is that they're part of a belt of areas that go around the middle of the brain around here. And here as well, we also have areas that are trying to instruct the motor cortex and tell it what to do. And there are yet more of them up here. I'm not going to go into all of their names, but I'm going to tell you about some of the ways in which some of these other areas work, because the way in which they tell the motor cortex what to do is quite different. It maybe works in a way that's a little bit unexpected. So here we've got a human brain, and what we're doing is we're looking at activity in that brain using a neuroimaging tool, like the sorts of tools that Claire works with. So we're going to put people in an MRI scanner, record when their brain is active, when they're making decisions about what they're going to do. And this blob of activity here, this is here because people are deciding about what to do next. And the activity that's shown here contains a couple of different signals in it. So we can pick out these signals. One of them's in red and one of them's in blue. And I'm not going to go into all of the details about how we pull out these signals, but it turns out that this brain area here it's trying to work out whether or not you should do the same thing as you did just now. One of these signals in blue is related to repeating what you just did. And the other signal here in red is telling you to change your behavior and to go and do something else. So probably the brain works a little bit like this because most of the time, for most of our evolutionary history, whenever an animal has learnt that doing something is good, it's probably a good idea to do it again a few seconds later. If something is good now, it's likely to be good in a few seconds' time and perhaps in a few minutes' time. And so there are brain areas that are not learning these arbitrary rules about what to do when you see a green light or a red light. There are brain areas doing that, but there are also these other ones that are just saying, do the same thing as you did just now. It worked just now, so do it again. And then sometimes there are signals in these same areas that are saying, okay, now it's time to switch, go and explore and do something different. And so that's been a fundamental way in which animals, all sorts of animals, have behaved for many millions of years. They've been built to forage for food, and they do that by cycling between periods of repetitively doing the same thing again and again and again. These brain mechanisms tell them to do that, and then occasionally they say, okay, that's not working anymore. Let's switch and go and explore and do something different. So we can see these patterns of activity in human brains. We can also see them here in a monkey brain. So here's a signal saying, go and do something different, or here's a signal saying, go and do the same thing again. And we've recorded these signals in an animal brain because what we were interested in doing is seeing if we could manipulate them at all. And we've developed tools here in Oxford using ultrasound that non-invasively change activity in these brain areas. And so you can change whether or not the animal will be likely to repeat the choice or to switch and do something different. So by manipulating things in this way, we as scientists are just interested in really checking our hypotheses about how these things work. But potentially as well, these could be tools that are developed outside of the lab for scientific research in order to look more at the human brain. Here's Elsa Ferragnan, who did some of that work with the macaque monkeys here in Oxford, now applying some of the same techniques to human participants in her laboratory in Plymouth University. So these techniques for exploring the brain are being taken out of the animal lab and being brought to use with humans and patients increasingly. So what this figure is showing you is we're looking at patterns of switching and repeating behaviour and showing that we can alter them with this stimulation device. Now, this brain area is also interesting because, as well as deciding whether or not to repeat a choice or go and do something different, it's also interested in working out whether or not you should be acting quickly or slowly. How fast should you act? So, as Claire mentioned earlier on, one of the fundamental things we need to know is some of the basic building blocks of what makes you decide to act. Well, one of the things we need to know is, should you act one way or another way? Another thing is, how quickly should you do it? And we know that this brain region also is interested in not just switching and staying, but in how quickly should you act. And it turns out that it knows this through interactions with a part of the brain down here called the basal forebrain. And this produces a chemical, a brain chemical called acetylcholine. And so what we think is happening as these two brain areas interact is the brain is going into one state or into another state. It's going into a state where it acts again and again and again quickly because that seems like a good thing to do, or it goes into a state where it starts to move more slowly. And again, these might seem slightly odd things for a brain to do, but they've been adaptive for most of our evolutionary history. If there's been something good and rewarding out there in the world, well, if you've managed to discover that, it's perhaps time to press your advantage and get more of it. If it's available now, maybe it'll be available again in a few seconds. So these mechanisms here control the transitions in our brain states as we move between periods of perhaps inaction to periods of frequent activity. The other areas that this brain region interacts with are down here. This is the dorsal raphe nucleus. This is the brain's own font of the chemical serotonin. So serotonin is one of the chemicals that is the target for a number of drug-related interventions. We have our own source of it here in this dorsal raphe nucleus. And this is also telling this singular area something about when to act as well. And what it's doing, we think, is it's saying, well, how good is your environment that you're in at the moment? So are you in a good environment? Are there abundant opportunities out there? Or are things scarce and rare? And depending on how your brain assesses the abundance or scarcity in the environment around you, it decides how urgent the need for action might be. So if you're in an environment with few and scarce resources, you better snatch anything that's going. However, if you're in a rich and abundant environment, then perhaps there's no need to be quite so impatient. And so we think this system here is controlling the way in which you act. Okay, so just about done now. So here we are back again with this motor cortex and premotor cortex. So what I've tried to tell you about is that the way in which we act is something that we can understand at a biological level. We know that it depends on this red area of the brain here, the motor cortex, but it doesn't do it by itself. It gets its instructions from other brain areas, and there are multiple types of instructions that are competing to get in there. Some of them are very rational things that we've learnt through experience, but some of them are these very ancient brain mechanisms for repeating what has been successful before, or switching and doing something different. What I haven't told you about is this area in white here. This is the prefrontal cortex, the area, the rather self-important area that Claire referred to earlier on. This is the area that's actually telling the premotor cortex what to do. And you can see that again, this is a primate speciality. Here it is in the human, here it is in the macaque, and sorry, mice and rats, there's not much going on here. So this is again another key aspect of what controls the way in which we make decisions, and it's maybe the thing that perhaps we can intervene in terms of the way in which we construct narratives ourselves about how our behaviour emerges, and how we might intervene and control that ourselves. Okay, so thanks very much for your attention.